Alright, so the second part of chapter 7, we're going to be talking about the cell membrane a little bit, and as you can see, we're starting to put the bigger picture together here. So we got to spice it up. Now, in order to spice this thing up properly, we got to take some advice from Emeril. You don't know who Emeril is? You got to watch more TV.
Grab a little pinch like this. Yeah. Okay, and you just like that.
So bam. Yeah, it's like that. Alright, so let's follow in stride with Emeril and spice the plasma membrane up. Again, it's called the fluid mosaic model. Fluid, because it's flexible and the components move within the membrane. The mosaic, because it is made of a ton of different things. Here we can see even more things. Looks like there is a cytoskeleton immediately beneath the surface. So we got our cytoskeleton down in here. Glycolipids attached to the surface. Carbohydrates attached to some of the proteins. And we have different types of proteins. We can see some of them here. We have peripheral proteins, integral proteins. There's a few others as well, glycoproteins up here. Now it's the proteins that determine most of the membrane's specific functions. In this image we see the peripheral proteins, which are bound to the surface of the membrane, right down here once again, as well as the integral proteins, which penetrate the hydrophobic core. Once again, the inner region here is hydrophobic. The integral proteins are also called transmembrane proteins, and are the proteins that contain those nonpolar regions holding them within the membrane. We can get a better idea of these hydrophobic regions by looking at a closer image of these integral proteins. And we can see the alpha helix regions of the proteins. Here's our alpha helix right down here. It's pointing to the inner part. These are the nonpolar areas. And remember that proteins are made up of amino acids, and that some of those amino acids are nonpolar. So we talked about some of the different types of proteins, but now some of the different functions that those proteins can do. Looking at image A up here, so yes, some of them are used for transport. We can see this solute using the hollow tube part of the protein to get into the cell. We can see this one over here is also used for transport, but this one is requiring energy. Some proteins act as enzymes, so we have enzymatic activity. Once again, enzymes breaking reactants in the products. Some are used for receptors or signal transductions. We have glycoproteins for cell-to-cell recognition. We also have intercellular joining, as well as attachment for the cytoskeleton and the extracellular matrix. So coming back to this image, we're going to be talking about the permeability of the membrane. As we discussed earlier, items can enter or exit a cell, and the cells must exchange materials with their surroundings. This exchange of materials is regulated by this plasma membrane. Also mentioned earlier, some things can do this easier than others. Nonpolar molecules, such as the hydrocarbons, can dissolve through the membrane readily, whereas polar molecules, they don't cross so easily. The atoms that cannot readily cross the plasma membrane will require the use of one of those transport proteins. So maybe one of these proteins is a transport protein. Another term sometimes given to those transport proteins are channel proteins, because they have a hollow channel. If you can think back to one of the earlier slides we looked at in chapter 7, we saw an image of a protein, I think it was on the first or second slide, for the potassium ion. And that had a channel that the potassium ion used to get into or out of the cell. These channels have a hydrophilic lining, which aids in an easy passage. Special proteins called aquaporins are used specifically for water. So water does not go straight through the plasma membrane because it's polar. It needs to use one of those proteins, the channel proteins, to get in, and the specific name of the protein for water would be an aquaporin. There's other proteins as well called carrier proteins, and these have binding sites for items needing to cross the membrane. Once the substance is bound to the protein will carry the substance across the membrane, changing the shape in order to do so. These proteins are quite specific, however, and will operate for specific items only, such as sugar. Kind of like the aquaporins that work specifically for water, that's about it. So now we're going to start talking about some of the things that cause or force items to cross that plasma membrane, either through use of the proteins or straight through the membrane. And the first one we're going to talk about is diffusion. Diffusion is one of the methods of crossing that membrane, and these items that cross in diffusion are typically called solutes, as they're dissolved or are soluble in water. These cross during diffusion without any energy needed by the cell, and since no energy is needed, the driving force is the concentration gradient, and the solutes are said to cross passively. Hence the name passive transport, which I'm sure you probably have heard in the past. Passive meaning no energy. So in short, solutes move across the plasma membrane from an area of high concentration to an area of low concentration, and we can see this in the image in the top left. The left side of the top of the membrane has a lot of molecules, a lot of solutes, and the right side has none. Think of the last time you were standing next to some dude who doused themselves with a heavy dose of Axe body spray. You probably got nauseous off the fumes. However, people safely standing 50 feet away still probably smelled those death fumes, but it was at a much more tolerable level because the vapors had a chance to spread out by the time they reached the nostrils. And as we can see in this image, diffusion is directional and it stops at equilibrium, which is what we see going on. Once again, high concentration, low concentration, it moves from high to low, so it goes down the concentration gradient passively, and it stops when equilibrium is reached. The next form of transport is called osmosis, and it's a form of diffusion, but it's specific to water. So since it's specific to water, it's given a unique name, and that's osmosis. Because it is diffusion, it is also another form of passive transport, which always strikes me as odd because, well, here's where we start and here's where we end up. Water goes up, which I always find kind of intriguing. So once again, water, it is a form of diffusion, and is trying to establish equilibrium passively. So water will cross a membrane, typically when solutes cannot cross the membrane, or if there's a very low amount of solutes, if any at all. We can almost think of this like the last time maybe your dog peed on the floor and you threw a paper towel on it. The urine or water on the floor moved from where there was more of it to an area where there was less of it. And when did it stop? Well, it stopped when the concentration of water in the paper towel was equal to the amount on the floor. This is the point where the paper towel is completely saturated. We see this going on in the top image in the curved type, in the curved tube. The left side has a low concentration. We can see that there's only a few solutes here, and on the right side there's a ton of solutes. Water moves from the low concentration to the high concentration through the membrane that they have represented right here. It does so passively, and this process is quite essential to plants. So taking this idea of osmosis and applying it to living cells, we have animal cells and plant cells that we see on our screen, top and bottom. Cells will be exposed to one of three types of solutions, and they are hypotonic, hypertonic, or isotonic solutions. What needs to be understood is the effects that each type of solution will have on a cell as a result of differences in those osmotic concentrations. To me anyways, isotonic is the easiest to understand because I know that iso means the same. So isotonic solutions are already in equilibrium with cells. So cells remain unaffected by these isotonic solutions. We see this in the middle image here, and for every little bit of water that enters a cell, an equal amount of water will leave the cell. So the cell is said to be normal, or in case of plant cells, flaccid. So basically what this means in an isotonic solution is that there's no tendency for water to enter the cell. Now the two more confusing ones aren't really too confusing. I like to use the phrase hypo-hippo to help differentiate the differences between these solutions. Now to do that, the hypo-hippo thing anyways, I know that hippos are really big, so hypotonic solutions will make cells get really big. And this is due to hypotonic solutions having a lower concentration than the inside of a cell. So if it's, keep in mind this is osmosis, guys, high concentration inside the cell, low outside the cell, water moves from low concentration to high concentration. In this case, water will move into the cell, which can cause cells to swell up. They'll get large, hypo-hippo, and possibly lyse or burst. In the case of a plant cell, this is called being turgid, which is an extraordinarily firm, hard cell. Now in plants, it says down here that this is normal, and if you think about it, that's kind of good, because plant cells, they want water going into them. They need the water for photosynthesis. But you know, with an animal cell, it's going to be a pretty sweet Mortal Kombat finishing move. And the last one is called a hypertonic solution. Hypertonic solutions causes a cell to lose water. This is called crenation, and can lead to the death of a cell due to dehydration. In plants, this is plasmolysis. This is because the hypertonic solution has a higher concentration than the inside of a cell. Keep in mind that water moves from low concentration to high concentration. We can see that this red blood cell, I don't know if you knew this, but these are red blood cells, are getting shriveled up as a result of the loss of water. Or in the case of plant cells, they're getting really shriveled up, or as much as they can anyways, or plasmolyzed. This will conclude part 2 of chapter 7. We'll pick up with proteins and facilitate diffusion and other forms of movement across the cell membrane when we get to part 3.